Alright, this next section, section 3 for chapter 10, looks at antimicrobial resistance. And there are just two objectives for this, the two ways that microbes acquire antimicrobial resistance, and then looking at the different cellular or structural mechanisms that microbes use to actually achieve this. So, drug resistance is an adaptive response in which microorganisms can begin to tolerate an amount of drug that would normally be inhibitory. And this can occur very slowly over time, or it can be something that happens abruptly. And how this occurs in kind of the big picture is through genetic versatility and adaptability of the microbe populations. So this could actually be an intrinsic characteristic, or it can be acquired. So with intrinsic, right, there's the idea that it always had the ability to do something, it just didn't know it had the ability. And we can probably apply that to our own lives, thinking that we couldn't do this. We couldn't do a particular activity, a different task, and then under severe stress or just building up the confidence, we find that we can actually achieve that. And the way that organisms do this is through genetic mutation, so acquiring genetic mutation, and then simply being able to adapt. So adaptability and genetic mutation are the two big categories in which microorganisms can develop resistance. All right, there you go. Objective number 10, adaptation and genetic mutation. So this is kind of a fun figure to look at because it shows the different ways that through adaptation or genetic mutation, bacteria can have drug resistance. All right, so there's 5 examples here. The top one is looking at the ability of an organism to secrete an enzyme that breaks down the drug, the medicine. This example, and this is going to be one of the major examples of this type of drug resistance, is penicillinase. So the penicillinase is an enzyme, given the ACE in the name. It's an enzyme that will degrade penicillin. And so here's the structure of active penicillin that gets into the environment of the bacteria, and the bacteria secretes then penicillinase outside of its cell, generally, and then it degrades or breaks down penicillin into an inactive form. And then the penicillin is no longer active, and the bacteria can continue to grow and amplify. All right, then the other one, or the next one, is permeability or uptake of the drug into the bacterium is decreased. And how does that occur? Well, if you look at this drug receptor here, all right, just the receptor that the drug binds to, and then it gets into the bacterium, and then it can kill off the bacteria. Well, the bacteria realizes this, and what it can do is through genetic mutation and adaptation is alter the receptor. And so now this receptor is more flat. The drug hits it and just simply bounces off. And then the drug is no longer able to get into the host cell or into the bacterium, and the drug is not effective. The third one, or the middle guy, and this is my favorite, all right, these are the development of drug pumps. And the drug gets into the cell by binding to the receptor, and it kills off the cell. However, some bacterium have been able to produce a drug pump. And what this drug pump does is simply, once the drug gets inside of the bacterium, it pumps it right back out into the environment. And then the drug is no longer effective. And the kind of the example that I use in class is the old Wile E. Coyote and the Roadrunner cartoons, where Wile E. was always trying to kill or capture the Roadrunner, and sometimes he would use the little grenades. And he would throw the grenade at the Roadrunner, and the Roadrunner would kick it back at Wile E. Coyote, and Wile E. Coyote would be the one who got blown up instead of the Roadrunner. And there's other examples of that in the cartoons from the 1980s. But that's what happens. Right? The drug is intended to kill off the bacterium cell, but the bacterium cell pumps the drug out of its cell before it's able to be effective. Right? Then the fourth one is that binding sites for the drugs are altered. And it's hard to see here, but right here, this is a triangle. The drug is a triangle shape, and it's going to inhibit the ribosome activity. And there's a little triangle space on that ribosome where the drug sits there, binds, and the ribosome is no longer able to function, and then protein synthesis decreases. But over here, instead of there being a little triangle shape right there, it's now circular. And so this drug, still being a triangle, is no longer able to bind to the ribosome to inhibit protein synthesis. You have the binding of the two ribosome components, the different subunits, and now protein synthesis can occur, and this drug is no longer effective. The last one is finding different metabolic pathways that can work when one of the drug pathways is stopped. So here you have this metabolic pathway to get to the product. This product is needed in order for metabolism of the bacteria cells, for them to grow. The drug is going to block this part of the pathway, and so then this process is stopped and bacteria growth can stop. To get around that, the bacterium cell just figures out a different direction. All right, still to get to the same final product, and now the cell can continue to grow. All right, and the last slide of this chapter, or this section, is looking at how we can actually select for drug resistance in our own body. And what you see at the top is bacteria cells that are being grown in a test tube that do not have any antibiotics. And they are grown on the surface of a petri dish. And the white colonies here are the colonies that are made up of bacteria that are sensitive to the drug. And then the red colonies, these two, represent bacteria that are actually resistant to an antibiotic. The next idea then is, if you grow these same bugs here in a broth that has antibiotics, then over time, just in a short period of time, an hour here, all of the bacteria that are sensitive to the drug die off, and these two are left. You just have the red guys. If you continue this process for, say, 6 hours or 24 hours, all of the white bacteria are going to be killed off because of the presence of the antibiotic here. And what you then have is, because you've given it plenty of time to replicate, so now you have a whole lot of these red bacteria which are drug resistant. And so if you have a couple of bacteria cells that are resistant to a drug, and you take that drug, then you kill off all of the sensitive guys, and you leave a lot of room then and extra nutrients for the resistant guys to grow up and expand. And now you have an entire intestinal tract, right? That's dramatic. But you have a lot more drug-resistant organisms in your body than what you previously had.